Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. If you like this deep dive and find these insights helpful, please hit that like and subscribe button and share it with friends and family who are curious about the cutting edge of biological research. It truly supports the Sans Science Experience and helps us keep unpacking these dense papers for you. Today, we're plunging into a study that provides, well, potentially a critical missing link in the biology of aging.
Yeah.
We're looking at new findings from Drosophila, that's the common fruit fly,
Right.
to investigate this connection between chronic inflammation, healthspan decline, and problems with mitochondria.
Yeah, it's a fascinating area. For a long time, we've kind of treated chronic inflammation and aging, inflammaging and mitochondrial breakdown as sort of separate issues. Hallmarks of aging, yes, but maybe not directly linked mechanically.
Okay.
But this research offers some startling clarity. It suggests the bridge between them is actually rogue mitochondrial DNA, mtDNA.
Oh, it's DNA.
Yeah, DNA that escapes from the mitochondria. The study shows that when this mtDNA gets out into the main cell fluid, the cytosol, it activates the innate immune system from inside the cell. And that seems to drive the inflammation that shortens a healthy life.
Wow. So this rogue DNA idea, why would our own mitochondrial DNA suddenly be seen as a threat just because it's in the wrong place in the cytosol?
Well, it really goes back to deep evolutionary history. Mitochondria, remember, were originally free-living bacteria that got incorporated into our cells billions of years ago.
The endosymbiont theory, right?
Exactly. And their DNA still has features that resemble bacterial DNA. So when it spills out into the cytosol, the cell's surveillance systems basically see it as foreign, like genetic material from an invading microbe.
Yeah.
It recognizes it as what we call a damage-associated molecular pattern.
So it triggers an immune response.
It's a strong one, an innate immune response. And the core idea of this study was to test if controlling that specific trigger, the response to the escaped mtDNA could actually extend health span. That's a pretty specific therapeutic angle.
Our ancient history biting us back. Okay, so let's dig into the evidence. How did they actually show this DNA leak is a real problem in older flies?
They did some careful work. They looked at aging Drosophila tissues, focusing on places that use a lot of energy, like the flight muscles in the thorax and specific neurons in the brain, the mushroom body neurons involved in memory.
Okay.
And by carefully separating the cellular components, they measured the amount of mtDNA, specific genes like COI and ND2, found just in the cytosol. And they found a really striking age-related increase, much more cytosolic mtDNA in 45-day-old flies compared to young 10-day-old flies.
So it definitely accumulates with age. And the link to inflammation, what specific pathway gets switched on?
It directly activates a pathway called REL-NF-BB. This is a master regulator of inflammation, really conserved across species, including us.
NF-kappa B, yeah, heard of that one.
Right, and the confirmed activation by showing that key immune genes, like ADA and TOTA, think of them as the fly's antimicrobial defense signals, were significantly upregulated in the older flies with more cytosolic mtDNA.
Okay, so the picture is older flies, leaky mitochondria, more mtDNA in the cytosol. This flips the NFE switch, leading to chronic inflammation and generally poorer health.
That's the core pathology they established.
Which logically leads to interventions. You mentioned they tried two main angles, like cleaning up the mess versus turning off the alarm.
Exactly right. Strategy 1, deal with the source or the DNA itself. Strategy 2, deal with the response to the DNA.
Let's start with cleanup. If damaged mitochondria are leaking, does fixing them help?
Mitophagy, right? That's the cleanup process.
Yes. Mitophagy is the cell's way of recycling damaged or dysfunctional mitochondria. So they tested if boosting mitophagy by overexpressing key proteins involved, like Parkin or DRP1, would make a difference.
And did it?
It did, significantly. Even when they started the intervention in middle-aged flies around 30 days old, inducing mitophagy effectively reduced that age-related buildup of cytosolic mtDNA.
But wait, if you just make the mitochondria healthier overall, maybe the flies get healthier for other reasons, not just stopping the leak. How do you isolate the effect? That's a crucial point. And that's why they looked at the inflammatory markers again. They found that boosting mitophagy also damped down that NF-key immune response. Those ADA-A and TOTA levels went down.
Ah, okay. So fixing the mitochondria did specifically reduce the signal that was causing the inflammation?
It strongly suggests that stopping the leak was key to stopping that specific inflammatory driver.
Okay, fascinating. But what if fixing all the mitochondria perfectly is, Hard. What about just getting rid of the rogue DNA that's already floating around? The second part of that cleanup strategy.
Right, bring in the demolition crew, essentially.
Yeah.
They used Nuclease's enzymes designed to chew up DNA. They focused on two. One called DNase II, which works broadly, kind of everywhere inside lysosomes, and another called SID, or stress-induced DNase, which they specifically targeted to neurons.
Because the brain aging aspect was important here.
Very important. And the results of just vacuuming up that loose DNA.
Yeah.
Both approaches, boosting DNase II everywhere, or SID just in neurons, significantly cut down the amount of cytosolic mtDNA found in the aged flies. The DNA got degraded.
And the effect on the fly's health?
Pretty dramatic. It wasn't just one thing.
Okay, break it down for us. What improved?
Okay, 4 key areas. First, lifespan. Both interventions extended the median lifespan. Flies live longer. Second, gut health. They use this thing called the Smurf assay.
Hey, Smurf assay.
Yeah, basically they feed the flies blue dye. If the gut barrier is leaky, which happens with age, the fly turns blue. The Nuclease treatments significantly delayed this Smurf phenotype, meaning better gut integrity.
Gut health, right, always linked to aging. What else?
Third, cognition. Better memory performance in learning tests, specifically olfactory aversion. And fourth, physical activity. The flies were more active spontaneously during the day and had better endurance in climbing tests. They were just more vigorous overall.
Wow, so cleaning up that stray DNA improved lifespan, gut, brain, and mobility. And I assume it also turned down the inflammation signals?
Exactly. They confirmed that NF key signaling was dampened. Less of the active form of the REL protein, REL49, got into the nucleus, and those ADAT and TOTI gene levels dropped. It really suggests that managing this DNA debris is crucial.
It really does. Okay, so cleaning works, but you mentioned a second, maybe more nuanced strategy, not cleaning the DNA, but changing the response to it.
Yes, targeting the sensor itself. This focused on a protein called EYA, which stands for eyes absent.
EYA, what's its job in this story?
They found EYA levels shoot up in the heads of aged flies. And crucially, it acts as a partner for NF key. It seems to be involved in sensing that cytosolic DNA and helping to activate the whole NF key inflammatory pathway. It's like a critical amplifier in the circuit.
So the intervention was to turn down EYA specifically in neurons.
Right, using a genetic knockdown approach just in the neurons of aging flies.
Okay, here's the key question then. If EYA is downstream, detecting the DNA, what happened to the amount of DNA when they turned down EYA? Did it get cleaned up somehow?
This is where it gets really interesting. No, the neuronal EYA knockdown didn't change the amount of cytosolic mtDNA accumulating in the old flies.
Wait, so the DNA was still there, floating around?
Still there, accumulating just like before.
But then how did it help? Isn't the DNA the root problem? Or is it really the inflammation that causes the damage?
That's precisely the question this experiment tackles.
Yeah.
Despite the DNA still being present, just reducing the EYA sensor function successfully turned down the REL-NF key activity. Less inflammation.
Okay.
And remarkably, this also significantly prolonged lifespan and improved those key health span markers, climbing, gut integrity. It strongly suggests that the major damage might be coming from the inflammatory overreaction to the DNA, perhaps more than the DNA itself.
That's a really important distinction. And you said they focused EYA knockdown in neurons. Did that have specific benefits for brain aging? Because neuroinflammation is such a huge issue.
Huge benefits. They documented slowing of 4 distinct hallmarks of brain aging just by this neuronal EYA inhibition.
4? Okay, what were they?
First, synaptic aging. They saw it suppress the age-related increase of a key synaptic protein called Bruch pilot, or BRP. Normally, changes in BRP are linked to memory decline, so keeping it more stable suggests healthier synapses.
Healthier connections between neurons. Makes sense. Second
Was cognition, which we touched on the improved memory in that olfactory test. Consistent benefit there.
Right. Third,
mitochondrial homeostasis. This is interesting. Knocking down EYA actually led to fewer mitochondria overall in the aged brains, but the ones that remained were healthier. They had significantly better membrane potential, measured by TMRE staining. Think better charged batteries.
quality over quantity for mitochondria, maybe.
Seems like it. And fourth, improvements in cellular housekeeping, autophagy and proteostasis. They saw increased activity of autolysosomes, meaning better recycling of cellular junk, and smaller protein aggregates, which usually clog up aging cells.
So hitting this one sensor, EYA, in the brain, sort of cascaded down to improve synapses, memory, mitochondrial health, and even protein cleanup. That's pretty profound.
It really is. It suggests this pathway is a major node controlling neuronal aging.
Okay, let's synthesize this. We have 2 successful strategies here. 1, Clean up the rogue mtDNA either by fixing mitochondria via mitophagy or by using nucleases like DNase II or SID. 2. Leave the DNA but turn down the alarm system by inhibiting the sensor EYA. Both routes lead to less inflammation and better health span.
That's the big picture. Exactly. It shows that this pathway cytosolic mtDNA activating NF-Cu via sensors like EYA is a really critical driver of age-related decline, at least in flies. And importantly, it offers multiple potential points for intervention.
And the paper hinted at why simply turning down the inflammation via EYA might even help the mitochondria that are still leaking. That seemed a bit paradoxical.
Yes, the discussion section proposed a really neat feedback loop idea. It goes back to what NF-key activation does.
Which is?
It ramps up the production of antimicrobial peptides, AMPs. These are molecules designed to kill bacteria.
Okay, so what's the connection to mitochondria?
Remember the evolutionary origin. Mitochondria share structural similarities with bacteria, especially their membranes. The hypothesis is that chronically high levels of these AMPs churned out because of the constant NF-B activation from the mtDNA might actually cause collateral damage to the mitochondria themselves.
Whoa, so the cell's own defense system, triggered by the leaky mitochondria, could be further damaging other mitochondria, a vicious cycle.
Potentially. So by inhibiting EYA and turning down NF-E, you reduce the levels of these AMPs, and that might inadvertently protect the remaining mitochondria from the cell's friendly fire, helping maintain their health even if some leakage continues.
That is a fascinating final thought. The idea that dialing back our own ancient immune responses might be key to protecting our cellular power plants as we age. It really reframes how we might think about tackling inflammaging. Thank you. This was a really insightful deep dive.
My pleasure. It's exciting work.
And thank you, our listeners, for joining us for this investigation. Don't forget to like and subscribe to the show for more deep dives into the world of science. This has been the SAN Science Experience. We'll talk to you next time.